My name is Daniel Saucier. I'm a PhD candidate at the Centre de Formation Médicale du Nouveau-Brunswick here in Moncton. And I'm here today to talk about some of the work I've done over the course of my master's degree, which I've completed, and some of the work I'm doing currently for my PhD. So the title of my talk is From Small Molecules to Population Level Research, using lab techniques and epidemiological methods to better understand amyotrophic lateral sclerosis. So a bit of an overview of the presentation. We're going to be talking about quite some interesting interdisciplinary projects between clinicians and researchers that collaborate together. On a first axis, we're going to talk about the diagnosis of ALS, working on that. So there's quite a few diagnostic challenges with this disease. I'll also introduce the disease for those that don't know what ALS is. We propose some potential biomarkers that might help fix these diagnostic challenges. And obviously we'll look at the results. So you get to see a lot of cool lab techniques for this first portion. And then for my PhD, I'm now focusing more on the etiology itself of the ALS. So our research problem is, why is there so much ALS in New Brunswick? We'll elaborate more on that later. We're going to look at the surrounding environment, which might be a source that explains the potential risk factors. And evidently, you're going to get to see a lot of epidemiological methods, as the proposed way we're going to accomplish this research goal is by using a longitudinal study. So what is amyotrophic lateral sclerosis, known as the acronym ALS? Well, if we break down it word by word, it simply translates out to being a morbid hardening of the nerve cells in the spinal cord that results in muscular atrophy. So as you can see below, a healthy motor neuron, it should be able to effectively communicate with your muscles of the body. So when you say, hey, I kind of want to flex my arm, it should have no problem transporting that signal. However, when motor neurons, which are mainly the things that are affected by ALS, they tend to deteriorate. As you can see, there's less insulation occurring, there's not as many synapses at the end. Unfortunately, we're going to lose control of the voluntary control of our muscles, which will eventually result in a progressive paralysis. The symptoms of ALS are quite varied, but at onset, we tend to have two clinical forms. The spinal form usually starts with simple muscle weakness. Maybe people are complaining that they're a bit more clumsy than they used to be, falling over their own feet, which can make it also difficult to walk. For the bulbar clinical form, usually the first complaints might be they notice that their voices is starting to change a bit, or they have difficulty swallowing. As the disease progresses, though, it tends to blur the lines between these two forms. And there's about 2,500 to 3,000 people that live with this disease currently in Canada. Unfortunately, most will die within 3 to 5 years after the onset of symptoms, as many will slowly have difficulty breathing, which is often the eventual cause of death. Treatment at the moment, there's nothing that's curative. There is 2 approved drugs at the moment, riluzole and edaravone. However, they only prolong life by a few months. There's many clinical trials going on, but unfortunately, the blood-brain barrier, which kind of divides the blood from the brain, is a big obstacle for having success in these trials. Another one, and probably more important to the patients, is unfortunately a lot are excluded if their disease onset has been more than 2 to 3 years ago, as this is often used to optimize ongoing trials. So a lot are excluded if they can't be diagnosed quick enough. The care provided by a diverse team of specialists is another solution at the moment besides pharmaceutical treatment. This care may prolong the survival and even improve the quality of life of ALS patients. However, when diagnosis is delayed, even by a couple months, this can significantly decrease the benefits that are associated with such teams. There's a lot of diagnostic challenges, which is why I'm hinting on diagnostic delay. There's no single test that exists for a definitive diagnosis of ALS. Clinicians often have to rely on clinical criteria and their observations to deduce if a case is indeed a true form of ALS. So usually that involves a neurologist, a physiatrist, or ALS specialist who might look to see using like an EMNG to see if there might be some upper or lower motor neurons that aren't quite working the way that they should be. We also have the ALS FRS. It's a rating scale. Although it's not used for diagnosis, it does help with tracking prognosis, which can help paint a clearer picture. Basically, we ask some questions regarding like their function to be able to speak or swallow. Score of 0 means no function, and 4 means full function. So you can monitor the progress of ALS using this, and it's also another way to indicate how a clinical trial is going on. However, patients, they often present with a lot of symptoms. It's a very heterogeneous disease. And that's why they often get mistaken for other diagnoses before they ultimately get the right one. In fact, the rate of misdiagnosis is believed to be anywhere from 27 to 61%. And the diagnosis is often delayed by a minimum of 1 year. You're very lucky if you get diagnosed before 1 year. And they'll often have to see up to 3 doctors on average before it's finally confirmed. So biomarkers, a measurable disease indicator, that's a bit more like a concrete number, that would definitely be necessary and would help facilitate the diagnosis of ALS. So Research Axis 1, diagnosis. The clinicians are mentioned early diagnosis is important, and our collaborator researchers are saying, hey, well, biomarkers would then be needed as a good solution. So Horizon, Vitality, Université de Moncton, and the awesome team at the Institut Atlantique de Recherche sur le Cancer got together, and what did they come up with? MicroRNAs and extracellular vesicles. Now, great, we got a solution, but you're probably asking, what are microRNAs? What are extracellular vesicles? And more importantly, why do we even care about them? Well, first of all, microRNAs are potential biomarkers. What are they? Well, if we look at them, we got a nice picture of them here, encapsulated in green. It's a nice little small chain of different nucleotides. So the stuff that like makes up our DNA, for example. It, on the contrary, is an RNA, and it's a special type of one. It's a non-coding RNA. What that means is it won't become a protein. Instead, it will just stay as is, but not for too long, because eventually it will separate into 2 different branches. And these branches, they're cool. They can link on to messenger RNA. And when it gets with a complex, it will actually stop them, these messenger RNAs, which are eventually supposed to become your proteins that do all the functions in your body, it stops them from being formed. So evidently, microRNAs can definitely regulate gene expression, which means they can have an impact in the body. But probably more importantly, why they're so popular as potential biomarkers is they found in various clinical samples, including the blood. They're also found in extracellular vesicles. And why are we looking at them in ALS? Well, it looks like that certain mutations in the genes that have been found in ALS have indeed potentially, they target different things that are responsible for the regulation of these microRNAs. So it is very possible that they are indeed dysregulated in ALS and would give a signature. So what are extracellular vesicles? Well, we can think of it as these little cell bodies that are excreted from the cell, and they contain a cargo of a bunch of different stuff. They may have some proteins, they may have some messenger RNAs, and obviously they have our microRNAs of interest. They're also found in various blood samples, including just like microRNAs in the blood. They have a physiological role. As you can imagine, if they're transporting different messenger RNAs, as they transport from one cell to another, those messenger RNAs, when they're in the new cell, they could be translated into proteins that have functions. Another good thing of them is they can also help with eliminating defective proteins out of a cell. However, as you can imagine, that means they can also have a pathological role. They could be responsible for transmitting the toxic proteins from one cell to another. And in fact, this has been potentially seems to be seen in some ALS models where it seems like indeed these extracellular vesicles may be playing a role in ALS propagation throughout the body. Now you're probably thinking, well, you're mentioning blood. But isn't this more like a brain and nervous system disease, the neurons? Well, fortunately, extracellular vesicles are released by cells of the nervous system. And what we can gather from the literature, it is pretty evident that they should be able to cross the blood-brain barrier, which means at least some of our signatures should actually be coming from the brain, or at least somewhere in the nervous system as well. So what is our hypothesis? Well, that there's a circulating signature of deregulated microRNAs that exist in the patients. And we're going to be looking at that one in blood as their circulating signature. These microRNAs, we think they also would underlie pathological mechanisms in this condition. Now that's all great, but how are we going to tackle such a complex task? Well, with the help of the interdisciplinary team of clinician researchers that we assembled. So first of all, for getting the samples, big thanks out to our two head clinicians, Dr. Colleen O'Connell at the Stan Cassidy, which is the provincial ALS clinic for all of New Brunswick, as well as Dr. Ali Marrero from the Dumont, who gets a lot of the French patients and those in the region of the Moncton Hub City. And evidently, we have a bunch of different clinical and health professionals that helped us as well to collect those samples. And obviously, we'd like to thank the ALS patients who donated, as I can imagine it's not that easy to do something such as that with all the symptoms that you're experiencing. And so great on them on being brave and very, very helpful in donating those samples. So ultimately, we recruited 14 patients and 12 controls who donated blood. They gave consent at visit 1. And although we have follow-up visits at each 6 months as the disease progresses, the talk for today and the samples that we're able to analyze during my master's degree are really just going to focus on visit 1. So what do you do with that blood? Well, you hand them over to our awesome blood fraction and expert, Sébastien Fournier. Big thanks to him. He's always available to make sure the second we arrive at that blood at the ACRI, that he's there to spin it in this fancy centrifuge machine so that the plasma can be separated from the blood. This is very important because there's a lot in the blood that can react very quickly and kind of dilute the signature that we're looking for. Once the plasma is divided from the blood, what's kind of cool is you can take this plasma and you can effectively capture the extracellular vesicles that are found in there, thanks to some technology that was developed by the ACRI team. In specific, obviously, Dr. Gosh, Stephen Lewis, Nicolas Cracpouret, and Rodney Ouellette. It's really fancy technology in the sense it's almost like a magnet to these extracellular vesicles. They're attracted to a certain protein that's found on the end of them. And if you add that VN96, which is their technology, that little peptide into that plasma, you rotate it for a bit and then put it in a centrifuge, all the extracellular vesicles will just form a pellet at the bottom of your tube. And then voila, you got your extracellular vesicles from your plasma. Well, you got the pellet, but obviously the pellet has much, much more than just microRNAs. As mentioned, that they may have proteins in them and other things. So what do we do to get just that RNA that we want, in specific the microRNAs? Well, you take that pellet, you put it in a solution that kind of destroys it, and you mix it up using a fancy scientific blender. You then add a bit of phenyl and chloroform, and you're able to do an extraction. You can almost kind of think of it a bit like oil on water, where there's two phases that don't like to quite mix. And what will happen is in the aqueous phase, you'll get your RNA, and in the other phase, you'll get your proteins. And the DNA, well, they just don't know where they want to go, so they just kind of hang in the middle. And that's great for us because after this extraction, you can simply put your pipette and take that aqueous phase on the top, and you got just your RNA. Now, obviously, there's all that chloroform, that phenyl, it's still all in there. Well, how do you get that RNA away from all those other chemicals? Well, you can kind of salt them out. You add a bunch of salt to your solution along with some ethanol, which is an alcohol. And all of a sudden, that's going to change the charges of our RNA. And, well, it's going to precipitate. And using a filter, you'll catch it. And then when you're ready and it's all washed, you can then put it in a elution sample. And voila, you've got some nice pure RNA to work with that contains our microRNAs. Big thanks to Annie Pierre for teaching me this technique and helping me with the samples as we had to process them very, very quickly. So to resume to date, we got our blood from our patients. We separated it to get the plasma. We added a VN96, which is kind of like a magnet to get our EVs. We broke up that pellet of AVs and we took specifically with an extraction just the RNA. Now we can finally quantify these microRNAs in the EVs using something called NGS, next gen sequencing. And this is a really cool technique because you can take your very, very, very small microRNA, you can add a little adapter to it, and it'll let you put a little barcode on it. It's like a little special sequence molecule that when it scans it, it knows, okay, this is so-and-so entry. So it's just like you're kind of at the supermarket. It's just scanning all the microRNAs in your sample going, okay, we got this one, we got that one, we got MIR1, MIR2, MIR3, because there's a bunch that exist in our sample. And it will identify the sequence because it's reading it nucleotide by nucleotide by using in the literature what microRNAs we know. Okay, if it's A, C, T, G, 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 A, well, that's MIR1, whereas a different sequence might be a different MIR. Big thanks to Simi. That's the NGS in case you're looking what it looks like. And she was the one who did all the work here and by no means able to learn such a specialized technique in such a short amount of time. And a big thanks to Dr. Gabriel Waschenberg, because that's a lot of data analyzed. He helped crunch the numbers and make it more digestible. So what were the results? Well, if we look at our NGS results, specifically just the ALS samples, here are the most frequent microRNAs that were in the sample, so the most present. As you can see, the MIR486 and a bunch of its alternative forms were all found in the sample, as well as LET7 and MIR26 were quite popular. Why is this interesting that for the microRNAs, these were the most abundant in the samples? Well, sure enough, in other studies where they took cerebral spinal fluid samples, so like maybe through like a lumbar puncture, they found these exact same 3 miRs were very abundant in their samples. So this kind of reassures us that at least maybe part of our signature that we're getting is indeed coming from the nervous system like we hope for, even though we're using blood. Now this is a heat map. When we look at NGS data, just kind of globally, we tend to use these to visualize what's going on. And this might seem counterintuitive for some, but in the realm of NGS, green means a microRNA is downregulated, whereas red means it's upregulated. So if we look at our patients here on the bottom, on the left, and our controls on the right, I don't know about you, but there's a lot more green in the patients and a lot more red in the controls. So what does that mean? For the most part, those microRNAs that are dysregulated, we could say that it looks like in the controls, for the most part, it's an overregulation, whereas in the patients, it's a downregulation, so less of those microRNAs being expressed. In fact, there's 22 that were downregulated significantly and 5 that were upregulated significantly. And this is even accounting for a false discovery rate. So you can imagine if you're testing a lot of different hypotheses, because each microRNA is a hypothesis of, is it different between the groups? This kind of helps adjust for that and it knows like, okay, yeah, this is a true indeed difference or very likely to be. Of interest among those 22 downregulated that have been seen in ALS models in the past, like cell models or animal models, is MIR9, MIR338, and MIR1246. On the upregulated side, MIR183 was one that was already seen in the literature to be upregulated. So it kind of reassures us that when we take actual patient samples, we're seeing what we've seen previous in the literature. So a big conclusion from this, a majority of microRNAs that were detected as differently expressed in patients with ALS, they're downregulated. And why is that such a big point? Well, it has been previously seen that when somebody has a decrease or total loss of microRNA expression in the brain, it leads to neurodegeneration. Also, it seems that the majority of microRNAs that are differently expressed in like kind of like spinal cord samples, where we kind of take a slice of like a lumbar sample, it looked like in those ALS patients, the microRNAs were mostly downregulated. So everything seems to be agreeing so far. And there's interesting mechanisms of how this microRNAs might have a role. When microRNAs are downregulated, when we look at cell models of neurons, it looks like neurofilament aggregations begin to form in the neurons. And this will eventually lead to the axons degrading because there's like too much of a block up here. And sure enough, MIR9, for example, it's supposed to rescue these neurons and stop the degeneration. But as we saw, MIR9 was one of the microRNAs that was heavily downregulated in the patients that we took blood from. So that would mean, unfortunately, maybe miR9 isn't able to do this rescue and might be a potential mechanism of how this degeneration is occurring. That also brings another point, maybe a future perspective. Maybe a potential therapeutic target would be to alter the biogenesis of microRNAs in these patients. MicroRNAs are decreased in the motor neurons of ALS patients, then maybe increasing them might help counteract that. And sure enough, enoxacin, which is a compound that seems to promote microRNA biogenesis, seems to improve challenges associated with the mice, what they were experiencing when we did ALS models of mice. And probably more interestingly is recently, there's actually been clinical trial recruitments to offer enoxacin to patients with ALS. So interesting to see what the results of those clinical trials will be. But our main question, did we find potential biomarkers that could help with the diagnosis? Yes, I'm happy to say we did. So this is a ROC curve. For those that don't know what a ROC curve is, basically you're looking at what is the sensitivity and a specificity of, like we could say, like a test or something that's going to help us divide people that have ALS from people that don't. And ideally, you can see this little line right here, and that's like all the space under the curve. That's why we call it AUC, it's area under the curve. You want it to be all the way over here to this corner, because if it's all the way to this corner, that means that your sensitivity, it's better. So that would be like 100% sensitivity. So if we said this person's positive once they have this much microRNA in their system, or this little microRNA in their system, it was indeed actually a true case. Whereas the other direction, it's kind of weird. You go the opposite way. You go from right to left, so more to the left being better. That means your specificity is too good. So okay, we got a good sensitivity with our cutoff, but also, we're making sure that our negative side is also working good, that we're not getting too many false positives, because you don't want that either. So for MIR15A, using the ROC curve, we can see that we get an area under the curve of 0.976. So what does that mean? Well, it means that the precision of this MIR for detecting those that have ALS versus those that don't would be 97.6%, which is fairly good. Now, that's fairly good, but can we improve? What if we use 2 MIRs as a way to detect? So like, you got to look at 2 MIRs at the same time to decide if someone is positive or negative for ALS. And yes, it does help increase the accuracy. In fact, it makes it 98.8% accurate. Now, a little caveat we should note is this is only with a small sample of patients, 14. So evidently, you'd probably want a bit more patience before we say this would indeed be the final accuracy. But it's still encouraging to see for a pilot study that it's indeed able to distinguish between the two very easily. And not just that, with prognosis. So by dividing our sample by those that are considered early stage ALS versus late stage ALS, so using their ALS FRS score, as mentioned, that kind of lets you know what is their level of function in. When we divide it, we notice that we're actually able, with 84.4% accuracy, to say who is in an early stage versus a late stage using MIR 193A. So that might be a great way to complement clinical trials to maybe see if there's an effect going on in the body before we even see the functional gains that are occurring. Now, evidently, as mentioned, again, take it with a grain of salt. We need a validation cohort, probably with a bit more, make sure these numbers still check out. And it'd be good to test those follow-up samples to see if these trends still hold, and maybe if our prognosis biomarker within the same individual continues to go down. Now, as mentioned, we also hypothesized that the microRNAs might underlie pathological mechanisms that might be linked to ALS. Sure, it's nice to have a marker that indicates you might have the disease or not, but it's even greater when you notice that it's actually maybe involved with the process of ALS. So maybe it's actually causing it rather than just, oh, those are the levels once you got ALS. So to do that, we have to rely on bioinformatics software, because it can be quite hard to test all the targets in a lab all at once. So to do that, we can use these programs called Target Scan Human and Miranda. And basically what they do is they have algorithms as well as information from studies that have already been done that say, oh, this microRNA, it should likely target this messenger RNA according to either past studies as well as their algorithms. Like, oh yeah, this microRNA matches up with this one, with this messenger RNA. And it's really cool because you use both at the same time, because obviously both softwares have different algorithms. So they're probably going to suggest different messenger RNAs, which ultimately become proteins. They're going to suggest different ones. And what you ideally want to find is a target that's this identical between the two methods. If it's the same between the two programs, well, then it's very, very, very likely that it's indeed a true match for those messenger RNA. And the ones that are matches, we usually underline them. So here's just an example with MIR144. We see that it, according to both programs, that NFE2L2 should indeed be targeted by that microRNA. Now, what does that do? Well, that would mean that it would regulate the levels of that messenger RNA, which eventually becomes a protein. And interestingly enough, this molecule is characterized for antioxidant properties. And sure enough, edaravone, which is a drug that helps treat ALS, is said to be an antioxidant mechanism. So in other words, it looks like indeed that is touching on a mechanism that could cause ALS. And deregulated activity in ALS models, it's been seen that indeed this pathway is deregulated. Now, as mentioned, this is just messenger RNA. Obviously, they'll eventually become proteins, and proteins can have a lot of different functions. We're probably more interested in knowing what actual biological processes and metabolic functions are affected in the body due to these deregulated microRNAs. So to do that, we need to find their gene ontology, their Go terms. And a program, a bioinformatic database called DAVID is very, very, very good at doing that. Basically, it'll match up all the different hits that you got for your messenger RNAs, and it will try to find out what processes are going to be affected most likely, or have been already confirmed. And as we can look in figure A, this is what biological processes are affected. And in B, we have the metabolic functions that are affected. And what I have highlighted in blue here is processes that seem to match up and all be similar together. We can see for a good percentage, the microRNA that were targeted, 20 all the way up as much as 40%, and obviously going downwards. Protein eupigination, positive regulation of phosphorylation, and protein kinase activity seem to all be heavily affected by the microRNAs that were identified as deregulated. That sounds like a bunch of mumbo jumbo, but what does that actually mean on a cellular and a more pathological level? Well, sure enough, it seems that proteins that are in a eupiginated form, a phosphorylated form, or are cleaved, like due to a kinase, sometimes that can lead to a pathological version, or like we could say a toxic version of a protein. For example, TDP43, depending on how much the deregulation of these activities are, depending on how it's phosphorylated, eucubinated, and cleaved, will actually have a great impact on diseases such as ALS and frontotemporal dementia, which is often associated with ALS. So a future perspective, well, seeing as we extracted the RNA, but we still have the DNA and the protein, because our approach was able to extract those too. They're still in the tube, we just didn't touch them yet. Maybe it'd be good to check and see if in the protein portion, is there those mutated forms, those toxic form of the proteins that are found in those patients? If so, well, that would help confirm what we saw bioinformatically and help confirm it in the lab. Now, one should always double check their measurements. So how are we going to do that? Well, we're going to use something called ddPCR. So we're going to take that same sample of microRNAs that we had, and we're going to run it in a machine to quantify the microRNAs in a different way. Now, this is a really cool technology. A big thanks to Dr. Jeremy Dubois for being, we could say almost like a supervisor when it comes to this technique, because it's very, very new. And I think there's great potential for this technique for eventually developing a bunch of different, or at least help developing a bunch of clinically diagnostic techniques with biomarkers. It's two machines. The first one is called our droplet generator. You take your little sample and you put it in oil, and then it like agitates it really, really, really quick to help kind of force it to mix a bit. And then you get a bunch of little droplets from your original sample. So you're almost like making a bunch of technical replicates all from one sample. You're getting like 20,000 droplets. And why is that great? Well, all of a sudden, we might have as little as 1 microRNA of interest in each droplet. And adding a little bit of markers. So we're almost adding like a barcode. And it's like, instead of like when you go to like, let's say like an airport, for example, and you have a metal detector and it detects the metal, here we're adding a special molecule that will only light up when it's the microRNA of interest. So then when we put it to the second machine, which reads luminescence, it will be able to say, okay, that droplet had a microRNA that we were looking for. Whereas if it doesn't have one, well, here's what it's going to look like. The ones that have one, they'll have a higher amplitude, whereas those on the bottom, they have the lower because they're not glowing up. You can then draw a line and say, yeah, I think that's where the division is. And that will tell you how many droplets had one versus not. And because there's maybe as little as 1 copy per droplet, that means you have a purely quantitative method. You can say how many copies, yes, copies of these little microRNAs there are per microliter of sample. That's very, very precise. And in theory, you wouldn't even have to run your sample again. We did for the fun of it a second time, and the results varied by maybe 1 or 2 copies in many cases. So this was a very good way to help to see if our results were similar to the NGS results we did. And sure enough, we picked a few that were upregulated, one that was downregulated, and one that should stay the same. If you look at our MIR15, which is supposed to be upregulated, because that's what we saw in NGS, the ddPCR noticed it was also upregulated using this technique. If we looked at some that were supposed to be, say, upregulated as well, like 185, it was also upregulated. Unfortunately, the one that was supposed to be downregulated, 181, it was just similar between the two, but at least it wasn't significant in the other direction. And the ones that were supposed to stay the same, well, they indeed stayed the same. So overall, we can say, using a different method to confirm our other results, the NGS data, yes, indeed, it should be accurate. So what can we draw from this? Well, we were able to characterize microRNAs. We should probably do a multiparametric approach to see the other things that we might find in there. Is there genetic testing that could be done on the DNA, pathological proteins? But at the end of the day, this is the first step among many to the contribution and development of a diagnostic approach using microRNAs. Obviously, we should do a validation cohort and maybe even test the mimic diseases to see if the signatures differ, because it would be kind of a shame if the mimic diseases had a similar signature to ALS. However, there's still a bunch of challenges we have with understanding the disease. And that brings us to our second axis, the etiology of ALS. So what causes it? Well, unfortunately, the cause is unknown. We know so little about ALS at the moment when it comes to this. A big contributor to that is most cases are considered sporadic, in fact, up to 90%. What this means is they have no known family history for the disease. So the cause is unknown. And although there have been a bunch of genetic mutations identified, in fact, as many as like 680, unfortunately, they only explain a very, very small proportion of cases. As you can see, all the white stick figures, that means we didn't identify a known mutation in the case. There's only a few that indeed actually have an identified mutation. And even the individual, if they have the same mutation, let's say both have SOD1, they could have completely different clinical presentations and completely different progressions, which is very interesting for having the same genetic mutation. So the hypothesis that the literature is coming to at the moment is maybe it's a complex genetic condition where there's small contributions, not just from genetics, but also environmental factors that could be explaining the cause of this disease and its heterogeneity. It's hard to study this disease because unfortunately it's very rare. We're talking like 1.9 cases per 100,000 people for global incidence annually. We're kind of lucky and unlucky that it might be easier to study this disease in New Brunswick. Lucky in the sense we can study it. Unfortunate that, well, it seems like, and this is really a patient-driven research question, a bunch of patients have been asking their clinicians, why is there so much ALS in New Brunswick? And sure enough, that resulted in Dr. Colleen O'Connell's team seeing if the patients were indeed correct, because they were talking on the phone and they found it weird that, hey, did you hear so-and-so was diagnosed? Oh, no way. So-and-so down the road was also diagnosed with the disease, and that shouldn't be happening. And sure enough, doing a spatial analysis between 2003 and 2013, the patients and their families were 100% correct. There was 187 patients diagnosed between this time frame, which is 2.5 cases per 100,000 people, and 92% of these cases were considered sporadic. So it's not just, we could say like it runs in the family per se. There was also 4 clusters of cases that were identified where the regions there was an excessive number of cases, more than there should have been, even adjusting for age and the population density. And they were found in Bathurst, Edmundston, St. Paul Parish, and Wilmot. And here we have a picture of Bathurst, for example, in the Beresford region. As you can see, each of those points, you're probably not going to believe me, it's a case. That shouldn't be happening. It's supposed to be a rare disease. So this really underscores the potential importance of environmental influence in the development of ALS. Looking at the literature, though, it would seem like when we look at potential environmental factors, according to a review that was already done, the only established risk factors would be male gender, where their exposures in an environment might differ slightly, and smoking. Other than that, military service, lead, and pesticides were probable risk factors where there's still a causal link, but there's not many cohort studies. Other than that, it's very, very, for the most part, unknown. There is really a need to better understand the role of non-genetic risk factors in the development of ALS, particularly environmental risk factors. So what not better way to start than do a more systematic review of the literature using PubMed and Scopus. And boy, was that a fun task. So we decided to do one for each of the classical elements. So earth, like rurality, air, so we're talking pollutants in the atmosphere, and water, so maybe proximity or the water quality, or what is the drinking water. So 1, 2, 3. And at the end of the day, what were the summary of each? Well, for rurality, it seems like there's many contradictory studies, but those that did find associations were the ones that looked more at stuff that was in the area rather than random designation of rural versus urban. It looks like industrial activities and water treatment facilities and agricultural areas might increase the odds of ALS, but more work needs to be done. For air pollution, it looks like exposure to diesel exhaust in your occupation had increased odds of ALS, as well as NO2 when it came to more the general atmospheric pollution in neighborhoods. And this is interesting because NO2 is actually one of the components of diesel exhaust. But what about other air pollutants like SO2, which is also found in diesel exhaust? Or what are the interactions? And then for water exposure, well, it seems like while the higher levels of certain minerals in drinking water was increased with an ALS incidence, these were only in 2 distinct populations. And while there is 3 cross-sectional studies that found proximity to lakes with harmful algae blooms might increase the risk of ALS clusters, there's been no follow-up long-term studies. So, and the buffer zones on these, we're talking proximity, we're talking maybe 4 to 30 kilometers away, which is quite far. So overall, most of the studies were unfortunately cross-sectional, and even then they relied on a questionnaire or interview to ascertain their environmental exposure, which obviously you're going to have recall bias when you do that. And that means at the end of the day, it would be important that additional studies have high resolution geospatial measurements that could objectively look what were the levels of pollutions prior to the diagnosis rather than at the moment. So we're hoping to do that with our study. So we hypothesized that the long-term exposure to factors related to rurality, air pollution, and water exposure would play a role in the development of ALS, and therefore maybe explain some of the regional differences we saw in New Brunswick, as well as the general observed increase in incidence across New Brunswick. Specifically, the level of urbanization, air pollution, and proximity to water for these cases in New Brunswick. How are we going to do it? A longitudinal study, specifically a matched case control study. And what's cool about this is rather than doing a regular prospective cohort study where you start with people that are exposed to a pollutant and those that aren't, and then you got to wait years and years and years until eventually they either develop the disease or not. We start with the cases in the control and work backwards. With a disease as rare as ALS, you would need a huge prospective cohort to have any chance of someone eventually develop an ALS. So this is the advantage of doing more of a retrospective nature one. And basically the idea is then you backtrack, and maybe we can go back as far as 1999 with the data we have. We look at all the exposure all the way up to the diagnosis and then see what the differences are. So maybe, who knows, maybe the cases were more often than not in industrial areas, where those that were controls, they tended to live in more natural areas. And in the matching aspect, we'll get matched for sex and age because, well, males are slightly more affected by ALS than females, and usually you're between 40 and 70 when you're hit by ALS. So this will help adjust for certain confounders that could exist. Now, while there's a big disadvantage for this, because when you do retrospective, you can usually only calculate odds ratios. So it's only the odds. So it's like there's more people that had this exposure in the cases than the controls, but you can't necessarily say it increases the risk. Well, fortunately, when a disease is rare, it's an odds ratio is very close to a relative risk, and you can consider it that. So it might have just as much power, this study, as a regular cohort study. So let's say we get a relative risk of over 1 for a certain exposure. That would mean there's an association, and you very well would be more likely to develop ALS if you're exposed to that exposure than somebody that wasn't. Our study population is going to come from the Stan Cassidy Center, which is the provincial clinic. We're going to use their medical records to identify their date of diagnosis, which will end our exposure period. We'll also get their Medicare number, so we can use MBRDT's various provincial and federal databases that have a bunch of other data we can use. Provisionally, we have at least 315 identified cases between 2003 and 2019. And with 2020 at it, we're even getting close to 350 potentially, which is crazy. Our controls are going to be coming from the citizens database that's available at MBRDT. It has all the residents from New Brunswick that have a provincial health card. So this is going to help us to match controls to the cases at a ratio of 1 to 4. And as long as they don't have ALS at the date of diagnosis of their matched case and they're alive, we're going to have similarly representative samples. Now, MBRDT, why I mention this so often, this is the main strength of our study that separates us from all other studies. Well, first of all, we got that citizens database. That's great for controls. It's going to make it easy for matching, help with statistical power a lot. We got vital statistics, discharge abstracts, physician billing. All that stuff has glorious demographics, and more importantly, a bunch of medical information that we can use to help confirm AL cases to make sure we don't have any mimic diseases, which has been a problem with some studies. We also have the postal codes of residents using these databases since 1999. So even the people that are from 2003, we could have as much as 4 years, and onwards we could have even more years of exposure that we could look before the diagnosis. And most recently, they got CANU data, Canadian Urban Environmental Health Research Consortium, which has longitudinal environmental database data with geospatial measurements at the postal code level. That's a lot better than 4 to 30 kilometers. So that's very precise. So this database will overcome the main obstacles of past studies on rare disease, statistical power improved, and we're not relying on questionnaires, so we might greatly reduce our information bias. For those that don't know CANU, it's cool. They have climate stations all across the country, as well as satellites that take spectral measurements and special algorithms that will adjust with these satellite measurements. So the further away you are from a climate station, they're still able to accurately calculate with their algorithms to high precisions what the levels of pollutants should be at every postal code. For level of urbanization, we're going to use the climate zone variables, which will class a given postal code and its surrounding 1 kilometer environment into one of 14 categories. And these are just a few examples below. We have paved, it might be sand, industrial, plants, dense forest. It's quite specific. And it's able to do this thanks to a bunch of measurements using thermal read-ins that might pick up on trees, for example, or might notice that the measurements are being affected by pollution, which would indicate industry. They also take into account known data in the area to make these assumptions. And using that, they can say the percent of pixels that align into one of 6 categories, which is kind of interesting, because then you could say, okay, sure, it's classified as this, but how much of that area has industrial influence? How much has natural, residential, water? And with that, our hypothesis is those that have more industrial in the area will have a higher quantity of ALS cases, whereas those that live in natural areas will likely have a slightly lower amount of cases of ALS due to maybe protective effects of the benefits of being exposed to healthy green spaces. For air pollution, we're going to use the air quality variables, specifically NO2 and SO2, which are the diesel exhausts that we saw in previous studies. It'll be interesting to see on a really long time scale in the general population if that indeed increases the risk. We think it will, seeing as it's what's found in diesel exhaust, for example. We're also going to look at ground level ozone, which when mixed with NO2, which seems to be responsible for ALS, and some UV and bad compounds, it will create ozone, which is ultimately the deadly byproduct which causes health effects. So we're proposing this might be the mechanism, maybe NO2 has its effect on the body, more deadly in ALS. And we're also going to look at fine particulate matter, because when you're exposed to that with SO2, it has been shown to have a synergy effect that will start neurodegeneration mechanisms, even at levels that are usually deemed safe. So that's kind of interesting. We're looking to see if maybe that interaction exists with the cases. And finally, water exposure. We're going to use the proximity water body variable from Canoe, which classes for each postal code the distance in meters to the nearest water feature within 5 kilometers. You heard me, in meters. That's pretty cool. Uses a coordinate system to accomplish that. And it can separate it and tell you what's the nearest ocean, reservoir, pond, or lake, water coast, tidal ward, or side channel, or canal. Our hypothesis is those that are still water will have a higher risk because algae blooms are more prone to form in those types of water, and that those that are exposed to SO in regions that have more SO2, it will likely increase this effect because SO2, the main way it gets out of the environment is through dilution in water sources. To do that, we're going to use logistic regressions that allow for repeated measures. So here we can see we have an ALS case would be labeled as 1, for example, whereas someone who doesn't would be labeled as 0. So that's our dependent variable. And then we can insert all our environmental predictors. And basically what that will do is we can see how do these environmental predictors affect the outcome of being diagnosed with ALS. And we can match them by grouping. So group 2 would mean that this ALS case and this control, we're comparing them directly one to the other, whereas another group would be another group of 1 case and 4 controls. And using time variable, so maybe this is 1 year before diagnosis, where this is at time of diagnosis, we can see how these environmental levels of pollution change over time. So that using mixed effect regression, this is how we're going to be able to model so many levels of data, repeated measures between individuals and groups, all the different subdivisions. And using a logistic function, we can then model the probability that 1 unit of change. So let's say maybe you go from the bottom quartile of pollution to maybe a higher level of pollution. What would be the increased probability of being diagnosed with ALS due to that increased level of pollution? And evidently, we could eventually interpret that as relative risk, but will let us know if there's indeed an association. And what is the effect size? So what do we expect will be the anticipated outcomes? Well, we'll advance our knowledge of environmental risk factors, which is really important given the high incidence in New Brunswick and the fact that we think it's estimated by the WHO that ALS cases will increase by 69% from 2015 to 2040. It might also highlight new avenues to explore in our ALS models to see if there might be new pathological mechanisms. So what mechanisms are being triggered by these pollutants? That might help to explain heterogeneity and maybe even develop more effective treatments based on what exposures they had. And obviously, it would help us to maybe strengthen public officials to be better equipped to implement new regulatory interventions to limit the amount of water contaminants or air pollutants there is, give stricter industry standards for pollution control. But probably the most important is we'll be able to answer the question of the patients, or at least maybe give some hypothesis for future studies of why are there so many ALS cases here in New Brunswick. If there's one message you should leave here with today, besides all the different techniques, is that great things happen when we work together. And that includes clinicians, patients, and researchers. So with that, I'd like to thank my supervisor, Pierre Morin, for my master's work, as well as a bunch of different other institutions for the funding. Evidently, the ACRI, as well as Dr. Colleen O'Connell and Adi Marrero, are clinicians, the Horizon Health Network, and the Réseau de Santé de Vitalité, for all the team there that helped with the collection and recruiting patients. And I'd also like to thank my PhD co-directors, Mathieu Bélanger and Dr. Colleen O'Connell, Zikuan Liu at MBRDT, who's collaborating us with the data, obviously the Stan Cassidy Center, and all people living with ALS, as well as all the numerous funding sources they have, in particular the MBHRF MSSU Award, which I'll have for the next 3 years, which will greatly help in answering this patient-oriented question. Is there any questions? I'd be happy to discuss as well.